In this session, we undertake the digital reconstruction of a fossil specimen derived from computed tomography data, provided in the form of an STL file. The objective of this workflow is to transform fragmented scan data into a coherent and anatomically interpretable structure. To achieve this, Blender 3D is utilized as the primary software environment for visualization, segmentation, and reconstruction. At the outset of this reconstruction process, it is essential to establish a preliminary identification of the specimen. This involves evaluating the overall morphology, the geological context in which the fossil was discovered, and the relative size of the specimen. Based on these combined observations, the closest morphological correspondence suggests that the specimen represents an avian fossil. The process begins by importing the STL file into the Blender workspace via the File Import menu. Upon loading the dataset, it becomes evident that the scan contains not only fossilized bone material, but also extraneous elements such as sediment, dust, and scanning artifacts. These non-biological components must be carefully filtered out to ensure analytical clarity and reconstruction accuracy. Following the import stage, the next step involves identifying a region of interest suitable for initial reconstruction. Rather than processing the entire dataset simultaneously, a selective strategy is employed. This involves visually assessing the available fragments and choosing a structure that appears relatively intact and morphologically distinct. In this case, an elongated bone is selected due to its structural continuity and identifiable features. To isolate individual fragments, all mesh components are selected and the workflow proceeds into edit mode. The Separate by Loose Parts function is then applied, effectively dividing the mesh into discrete objects based on disconnected geometry. This segmentation allows each fragment to be treated as an independent unit, significantly improving precision during selection and manipulation. The identification of the Tarrsometarsus is particularly important, as this structure plays a critical role in locomotion and weight-bearing in avian species. Its relatively robust and elongated morphology often makes it more recognizable compared to other skeletal elements, thereby providing a reliable foundation for reconstruction. Based on this anatomical interpretation, the reconstruction process is refined to focus specifically on the Tarrsometarsus region. Additional segmentation is performed to isolate only those fragments directly associated with this structure. These components are organized into a dedicated collection, while all unrelated elements remain hidden from view. With the target structure isolated, the sculpting phase begins. Sculpting tools are employed to refine the surface of the fossil fragments, smoothing irregularities and minimizing artifacts introduced during the CT scanning process. In addition, spatial adjustments are made to reposition disarticulated fragments, bringing them into closer anatomical alignment. Following the initial refinement, reconstruction proceeds with the introduction of supplementary geometry. A new mesh object is created, initially in the form of a cube, which is then elongated and aligned with the existing fossil structure. This object serves as a reconstructed extension, compensating for missing or incomplete portions of the original Tarrsometarsus. To enhance its manipulability, a subdivision surface modifier is applied to the newly created geometry. This increases vertex density, allowing for more precise shaping within sculpt mode. The reconstructed segment is then adjusted to match the curvature, thickness, and proportional characteristics of the original fossil fragments. Additional volume is introduced where necessary, and gaps between fragments are filled to create a continuous anatomical structure. It is important to emphasize that this stage involves a combination of empirical data and informed interpretation. While the CT scan provides a foundational reference, the reconstruction of missing regions requires anatomical reasoning based on comparative morphology, particularly with extant avian species. Once the reconstruction reaches a satisfactory level of completeness, visual differentiation is introduced through color coding. The original fossil elements derived directly from CT data are assigned a blue coloration, while reconstructed components are colored red. This distinction enhances transparency, clearly indicating which portions of the model are data-driven and which are interpretative. The workflow then incorporates the application of Blender's Rigify system. At this stage, it is necessary to distinguish between two uses of the term bone. In an anatomical context, bone refers to the fossilized skeletal structure being reconstructed, in this case, the tarsometarsus. In contrast, within the Blender environment, bones refer to elements of an armature used for rigging and deformation. The Rigify system is applied to the reconstructed model to create an armature framework. This enables controlled manipulation of the structure, facilitating adjustments in alignment, proportion, and orientation. Furthermore, the addition of an armature allows for potential future applications, including animation and biomechanical simulation. Through the integration of rigging, the reconstructed tarsometarsus is no longer limited to static representation, but becomes a dynamic model capable of demonstrating movement and functional behavior. This adds an additional layer of analytical value, particularly in studies of locomotion and comparative anatomy. In conclusion, this workflow demonstrates a systematic and anatomically informed approach to digital fossil reconstruction. By combining segmentation, sculpting, and interpretative modeling, it is possible to transform fragmented CT scan data into a coherent representation of the tarsometarsus. In subsequent sessions, the same methodology will be applied to other anatomical regions within the fossil dataset. Through iterative reconstruction, a more complete and scientifically meaningful model of the specimen can be achieved.